pleasure for me to announce the next speaker, Professor Didier Letourneur from Paris, the European NanoAthero project from the design to the clinical trials. Professor Letourneur, please.
Yeah, thank you. It's a great pleasure for me to represent this consortium. The next one is this one. Yeah, so this consortium, it's a large-scale project financed almost 6 years ago with 16 partners, 10 different countries, just finished this year. And I will present the key results in a relatively short time. So the whole idea was to develop nanosystem in the cardiovascular field and to demonstrate its feasibility in early phase 1 clinical trials. So the project was built on 4 lines of translational research, 2 focusing on thrombus and plaque, because thrombus is a key element in atherosclerosis, and plaque, of course, you heard that, a key component for atherosclerosis. And we were focusing on both imaging and treatment, but separately, not theranostic. So it was either imaging or either treatment. So we had at the beginning with the partners several types of nanosystems that were already in with good data, preliminary strong data, and we were having the idea to develop them and to screen them for the best application. So I will not go in detail of all of them, but just give you a few examples of what we have done until the clinical phase, because we have done 4 clinical trials with this nanosystem within the NanoAthero consortium. So the whole idea was with the partner to try to classify and to select the nanosystem, because each partner can try to develop this, but with the help of this consortium, we were able to select 7 key parameters. I will not go again in detail, but it's just published now, and it's just online this week in cardiovascular research. So we have defined these 7 parameters to select the right candidate nanosystem for the clinical testing. So among them, we develop them and prove that it's possible to go at least at phase 1 clinical trial. So the first example is a plaque treatment. For that, we use a nanosystem and a drug. And the drug was developed by Dutch teams, 2 Dutch teams, and we went until the phase 1 clinical trial. The drug is a prednisolone, a corticosteroid, loaded in a liposome. And it was first produced as a GMP compound by 2 partners, and with the help of another one for complement activation. So we had the preclinical evaluation of this GMP drug-loaded liposome, and then the clinical evaluation, and the results did prove that the liposome accumulate in human atherosclerosis macrophage. So the design of the clinical trial didn't prove that we have an effect, but we did found that the liposome accumulate in atherosclerotic macrophages in the plaque. So it's interesting to have a carrier. Now we have also to think about the drug and the best effect on the regime to deliver this in human. Another example, which was also provided by the clinical team at Amsterdam Medical Center, it's the feasibility of using iron oxide particles, so nanosystem, for patient, for the screening of patient. We heard that a key parameter is the screening of patient, how to select the right patient. So with a commercially available iron oxide particle, the team of Eric Strauss was able to detect a specific subset on different physiological aspects in the plaque in human as compared to activation, like which was produced by FDG PET screening. So it's interesting to develop this iron oxide particle, and this was exactly what one of the team in Erlangen, the Sion team, is currently doing with a new design of iron oxide particle coated with dextran, and they went until the 7th criteria of that, and now they are producing that at the GMP form for further clinical trials. So this is a good point for further clinical development. Another use of this program was to focus on thrombus. And we have 2 lines of development for thrombus, one for imaging, one for treatment. So we did a lot of work using a natural compound, which is a seaweed extract, which is a fucoidan, for the targeting of P-selectin. P-selectin is expressed in activated platelet, which is found in thrombus. And we were able to patent and to publish that we can target the thrombus. And it was interesting to target this thrombus and then to image it, because we were able to tag the fucoidan with several key elements for imaging. And I will not detail this, but we have several publications on this for the targeting system. And we have done that at the preclinical level in mouse and rat. So the idea was to develop in several imaging modalities. It's what we have done for ultrasound, for MRI, for SPECT. We are currently doing this for CT and for PET imaging, but still, so far, it was preclinical thrombus imaging. So we wanted to move to clinical imaging. So the next step was to prepare the GMP formulation of fucoidan. And it's what we have done with the collaboration of several industrial partners on subcontracting. So we were able to develop the GMP manufacture of this fucoidan. So we develop all the steps for the production of this macromolecular system. And then the next step was the regulatory toxicology dossier before going to clinical trial. With the help of the Bracco company, which was a partner in the NanoAthero project, we found that the maximum dose we tested was 40 micrograms per kilogram, so which was equivalent to 20 milligrams per patient, without any toxicity, and we wanted to inject only 40 micrograms per patient. So we have a wide range of safety for that. We had all the dossier. So the next step was to go to clinics. We prepare all the submission file. Of course, it's a lot of work, a lot of expertise to do that, but we then submit it to the French authority. We got the agreement by the French authority at the beginning of this year. We also get the agreement in March of this year from the Dutch authority, and then we launch the phase 1 clinical trial of this fucoidan, GMP grade for clinical thrombus imaging by SPECT imaging. This is one example of the patient. We did 9 volunteers in this phase 1 safety phase, and we detect only side effect on the usual uptake of this. So the phase 1 is now complete. We are now moving on to think that we could go further. We went from the seaweed extract, from the process of preparation of the seaweed extract until the phase 1 clinical trial. Now we have to move on. It will be outside NanoAthero project. I hope in another project. But we have now to go to phase 2, phase 3. Of course, it will still be a lot of work. So, of course, in the NanoAthero project, we also focus on many aspects. Of course, safety, toxicology, ethics, dissemination. I would like to ask you to go to the NanoAthero website. You will find many information of this. We have also several paper publication, and just I want to focus on one patent, which was also developed by the Medical University of Grasse, which is a bioinjury device which could be very interesting also for animal model or for better assay in this preclinical assay. With this, I would like to thank especially the European community for funding this project, all the partners and the collaborative work they have done, and also you for your attention.
Thank you very much for this interesting presentation of this data of NanoAthero project. Questions, please. We have time for 2 short questions. If there are no questions, I have one question to you. After the NanoAthero project now, what do you think is the most promising candidate for clinical intervention of atherosclerosis now?
Yeah,
to resume.
In fact, I cannot summarize in only one aspect because we have developed several type of modalities. It could be also for imaging. Imaging is very important point because if you can screen the patient before the treatment, it's a key aspect. So one key aspect will be the screening of patients. So iron oxide particle without any side effect is a very good point for the screening of patients and can be easily detected by MRIs, which is conventional imaging treatment. So pre-screening of patients could be a very important point. So for sure, we have to develop the GMP formulation and to prove it in clinical phase 1 trial. So pre-screening of patients. And then for imaging, as I said, for thrombus, it's already on the way. So we have to demonstrate in phase 2, phase 3. And for treatment, it's still more complex, but we have always to find the carrier, which is GMP, and the drug, which is already approved, and the right drug and the right carrier. So this is still much work to be done, but at least imaging for screening or for detecting thrombus could be already almost achievable.
Please.
Thank you for your talk. Congratulations on getting it to phase 1. That's always great. Was there any focus throughout this project on extracellular vesicles, either diagnostic or therapeutic?
No, in fact, it was already a large range of nano system. It could have been another system added to the list. We have already several, so we screen already some of them only to go for GMP condition on clinical phase. The translation on GMP formulation of extravesical, it's quite tough. Also, it's a key issue before selecting them for clinical phase. This is a question of quantity, reproducibility, characterization. So this is a main issue before selecting them for phase 1 clinical trial. Of course, it could be interesting, but it should be one project only by itself, I think.
If there are no further questions,